Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, as always, Ricardo Lopes, and today I'm joined by Dr. Günter Wagner. He is Alison Richard Professor of Ecology and Evolutionary Biology in the Department of Ecology and Evolutionary Biology and Adjunct Professor of Obstetrics, Gynecology and Reproductive Sciences at Yale University, and also Adjunct Professor of Obstetrics and Gynecology at Wayne State University. His research interest is the evolution of gene regulation as it pertains to the origin of evolutionary novelties. So, Dr. Wagner, welcome to the show. It's a pleasure to have you on.
Thank you for having me.
Okay, so how did gene regulation evolve?
Well, of course, those are two questions, right? The one is how gene regulation originated and how, once there is gene regulation, it evolves. And I think it's fair to say that we know more about how gene regulation evolved once it existed, like we have it in all organisms alive today. And so gene regulation can actually change in evolution on various levels, but the best studied area is where the control of transcription, meaning where the information in the genome written into DNA is transcribed into another molecule that's called a messenger RNA, and how and when this is happening, this is called this transcriptional control of gene expression. And the idea is that there are parts in the genome that determine whether proteins that initiate and continue the transcription of these genes get recruited to a particular part of the genome. And these so-called cis-regulatory elements, meaning DNA sequences that attract those proteins, they can change and thereby change their affinity to different so-called transcription factor proteins. So that's the main way how people think about the evolution of gene regulation.
Yeah, go ahead.
There is, of course, also another possibility that seems to be happening, but at a lower rate, namely that the proteins that control the expression of genes themselves change, namely by changing their protein properties and how they interact with other such proteins, and that would be called, transcription factor protein evolution. But admittedly, that is a process that is not as often found, but it does exist and may play a special role in the evolution of organisms.
Mm-hmm. And why is gene regulation important in evolution?
It's important because it turns out that a lot of the evolutionary changes that we observe or can infer for higher organisms like mammals and humans and so on are actually based on a change in the time and intensity of gene expression rather than the evolution of new genes. That also happens. The new genes arise and you get new proteins that contribute to development and physiology, but the primary mode of evolutionary change of animals and plants seems to be changing the pattern of expression, where it's expressed, when genes are expressed, to modulate developmental and physiological processes. So it is considered the major mode of the way how organisms change at the genetic level.
Mm-hmm. Are there evolutionary phenomena that we can understand better by studying gene regulation?
Well, yeah, that's and most of the morphological changes that happen can actually be traced back to changes in gene regulation. For instance, one where, for instance, digits change their morphology can be traced back to different patterns of gene regulation, meaning that certain transcription factor proteins are either expressed in one digit or the other, which then changes whether the digit is growing more or less, or getting thicker or having some other shape. And so that you actually find it all over the place.
Mm-hmm. And does gene regulation influence in any way how evolution unfolds over time?
Well, I would say it's one of the major ways how evolutionary change is realized. It's not so much an independent factor that sort of influences evolution. I think it is one of the major ways in which evolution unfolds and is happening. So in that respect, it is at the very core of evolutionary change.
Mm-hmm. But is evolution constrained by gene regulation in any way? I mean, the sort of paths it can take?
Well, I think that depends on the particular examples. Right now, I cannot immediately think of examples that would very strongly constrain evolutionary change, because it turns out that gene regulation, at least the transcriptional regulation, is in different parts of the body is controlled by different so-called cis-regulatory elements, so that you can change the gene expression in one part of the body without affecting other parts. Of course, as long as, let's say, fore and hind limb development is controlled by the same gene and the same cis-regulatory elements, the forelimb and hind limb evolution remains constrained, if you want, or correlated, and only if and when a gene regulatory logic or network is changed so that the forelimb and the hind limb is under the control of different genetic elements, then radical differences in the forelimb and hind limb can evolve, as we see, let's say, in chickens or in birds in general, where the forelimb specializes for flight and hind limb for running or just grabbing, or between in bats with forelimb and hind limb, and so on. So yes, there can be constraints depending on what the prior evolutionary histories of these characters, but there are ways of how these constraints can be overcome. For instance, by the origin of new gene regulatory elements like the one we are talking about. And the interesting thing is that completely new elements like this can arise from so-called transposable elements. Those are DNA segments that most likely come from viruses and that get integrated into the genome and can replicate. That means they make copies of themselves and insert in other parts of the genome. And if they happen to come into the neighborhood of a particular gene, they can create new opportunities for gene regulation. So as always in evolution, there's always an interplay between constraints and opportunities, right? If you don't have a lot of transposable elements in your genome, then the opportunities to get create new cis-regulatory elements, these gene switches, is less than if you have lots of them, which of course itself is a two-edged sword, because if you have lots of these transposable elements in your genome, they also can do a lot of damage and increase the mutation rate. And probably it's not an accident that mammals are known, for instance humans, that only 2% of our DNA in the genome actually code for gene products, proteins. The rest, about 50%, is still recognizable as former transposable elements or as transposable elements themselves. And maybe another 40% are not coding for proteins and not recognizable, but probably just degenerated forms of transposable elements. So, I mean, we are over 90% of our genome consists of these apparently parasitic elements, but we also, by having them, we also have the opportunity to innovate new ways of regulating our genes. So at the one hand, we pay a price for them by high mutation rate, maybe even cancer rates and so on. But on the other hand, it also gives us an opportunity, it's anthropomorphically spoken, but gives our lineage together with other mammals that have also genomes of similar size, many opportunities to come up with new ways of regulating our genes or the expression of our genes.
Mm-hmm. And how can we understand phenomena like pleiotropy and polygeny from an evolutionary perspective? Are there particular reasons why it is the case that we have genes that encode for multiple traits and traits that are encoded by multiple genes?
Well, pleiotropy, meaning that the same gene or the same gene locus plays a role in different parts of our bodies, I think is primarily a consequence of the fact that we are multicellular organisms. And so our body is composed of cells, and each of the cells has potentially exactly the same genes as the other cells. So any gene that is not explicitly turned off in one part of the body and only expressed in another part of body, so will have pleiotropic effects, meaning that if it mutates, it will affect all the cells in which this gene is expressed, right? So pleiotropy simply comes from the fact that we are multicellular organisms, and multicellular organisms means we are composed of cells that all have more or less the same genetic information. The question why we have polygyny, meaning that one and the same organismal trait is influenced by a large number of genes, is due to the fact that any recognizable part of our body, let's say the eye or the skeleton or the digestive tract, is the result of a long and complicated developmental process in which many genes play a role, right? And so if many genes are necessary or need to be engaged at different stages in the development, it means that mutations of all of those genes potentially influence this process. So polygyny is just a consequence of the fact that development is a complex process that needs the activity of many genes.
Mm-hmm. What is the relationship between development and evolution?
Well, if we talk about multicellular organisms, and I'm a zoologist, so the primary examples that I'm thinking of are animals. And of course, most of the biosphere is made up of single-celled organisms and bacteria and viruses and fungi and so on. But if I speak as a zoologist, and then evolution of animals is the evolution of the development of their bodies, right? So in each generation, each individual arises from an egg cell from the mother and a sperm cell from the father that contribute half of the genes that we have in our cells, and the body that we recognize as, let's say, as a human being or a dog or a chicken or whatever, is the result of an developmental process that comes from a single cell, namely the fertilized egg cell, making many cells. These cells then fold and organize themselves into different parts, and so viewing organisms as the result of development immediately leads to the conclusion that the evolution of these bodies has to be the evolution of the development of those bodies. So the evolution of bodies is the evolution of development. And in order to if we want to understand how bodies evolve, we need to understand how their development is evolving.
Mm-hmm. And what are evolutionary novelties?
That is a difficult question, and you probably will get as many different answers as you interview people. But I can give you my specific perspective on it, right? So let me go back a little bit and recapitulate what kind of processes we study in evolutionary biology. So at the one hand, we understand pretty well the evolution of what we call adaptations. Polar bears have a white fur for camouflage and also for thermoregulatory reasons, while in the temperate regions the bears have black or brown fur, and that's a clear example of adaptation. Or you have birds who use seeds of different hardness, then the shape of the beaks will match the mode of feeding, right? So you have, in both of these cases, you have a character, let's say the fur or the beak, and this body part then gets modified depending on what ecological constraints or necessities exist in the life of these animals, right? So that would be a typical example of adaptation. Most adaptations that we understand are modifications of things that are already there before the modification happens. The other kind of process that we study very intensely in evolutionary biology is the origin of species. That means that a population of organisms give rise to two different populations that then have a quasi-independent evolutionary history. That is what speciation means. And an important intellectual advance in the 20th century was to realize that the evolution of adaptations and the origin of species are actually two different kinds of questions, and actually driven by quite different population genetic mechanisms. So we have to so adaptation is not the same as the speciation. Speciation is not the same as adaptation. They can interact at certain levels, but those are fundamentally different kinds of processes. Now, if we come to innovation, my argument is that innovations are exactly those evolutionary processes that create a new body part that then can be subject to adaptive modification. For instance, the origin of hair. Hair only exists in mammals, right? And our ancestors that we have in common with the reptiles, we don't know exactly how their skin looked like, but we are pretty sure they didn't have hair, they didn't have feathers. Maybe they had scales, but even that is not certain. So that is one example of a novelty. And there are animals that don't have distinct anatomical eyes, and then eyes evolve, and then once you have an eye, you can adapt it to high light intensity, low light intensity, higher acuity of vision, and so on. Those are then the evolutionary modifications of a thing. So to me, an innovation is the origin of a new part of the body that then can acquire particular functional roles. Okay, and that again, like in the case of adaptation and speciation, to understand innovations is quite a different kind of question to answer. You have to pursue a different research program in order to understand innovations than you have to pursue in order to understand adaptations. So for the evolution of novelties, and I think maybe the simplest or the clearest example of innovations are new cell types. like light sensory cells that evolve from epithelial cells, from skin cells, for instance. In order to understand the origin of a new cell type, you have to understand the process by which the new cell type is able to control the expression of its genes differently from other cells, right? So either by changes to the transcription factors or new cis-regulatory elements and so on. And so how that in detail is happening, I think we don't know yet. We have good guesses that lead our research into it. But there are no simple and straightforward and well-established models yet of how that actually happened.
Mm-hmm. Since we started by talking about gene regulation, does it have anything to do with the evolution of novel traits?
Right. So my argument is that in order to have novel parts of the body, you can only develop new parts of the body if you find new ways of regulating genes that make this part of the body, right? So abstractly spoken. And, in the case of new cell types, clearly a new cell type only exists if the expression of genes that actually do physiological work like enzymes and extracellular matrix proteins and so on are expressed differentially. It means different from their expression in other cells, which means the origin of gene regulatory machinery that allows that is synonymous at the molecular level with the origin of novelties.
Mm-hmm. What is a complex organism, biologically speaking, and how do complex organisms evolve?
Well, again, that I'm not sure there is a consensus opinion about that, because at one level you could make a defensible argument that all organisms are complex, right? Even bacterium like E. coli, it has a very complex metabolic machinery and so on. But conventionally, what we mean by a complex organism is one that consists of many differentiated parts, right? So if you compare my favorite simple animal, which is called the Trichoplax adherens, which is looks like the name actually already tells it. It looks like a hairy plaque on the surface of an aquarium pane. And this organism is an animal, this is quite clear, but it doesn't have a head, it doesn't even have a gut. It only has a cell layer on top and a cell layer underneath. It can do digestion, but only by making a hollow structure temporarily, and then digests its food, and then it uses the same cells to crawl further. So this is a simple organism that has about 5 to 30 different cell types. No definite body axis, so there's not a definite front end and the hind end. There's no internal organ in there. And in contrast, a complex organism like a vertebrate, or let's take human as an exemplar. At the cell level, there are at least 500 different cell types, probably many more. This number of 500 is based on conventional histological or 19th century techniques, and with modern techniques it probably will increase at least by a factor of 4. Is my estimation. So compared to Trichoplax, we have at least 100-fold more cell types. We have a very definite body organization with a head and a limbs and a gut and a lung and a heart and all of the nice things that we learn in anatomy class. And the very fact that so-called complex organisms are composed of many different individuated parts is what we mean by a complex organism. And how it originates is, of course, by the same processes that we discussed before, namely the evolutionary novelties, the meaning that the origin of new individualized parts of the body. How that is happening at the molecular or population genetic level, I think that is still a very unsettled research question. And I can speculate about it, but you shouldn't take it as a definite answer, as a definite answer for this research program.
Mm-hmm. And what about functional specialization? What it is and how does it evolve?
Well, I mean, functional specialization is the mirror image of morphological specialization, because once you think about my examples of a differentiated complex organisms, all of the parts that I mentioned only exist because each of them is performing a specialized function. The brain, obviously, and the eyes for visual, and the ears for sound perception, and so on. So there is a certain complementarity between morphological or cell biological specialization and functional specialization. Now, that's sort of the first level answer to it. It's a little bit more complicated than that, because body parts, once they exist, can change their function, but still be the same thing. The limbs of tetrapods were initially structured to support the body on the ground, but they can also be used to eventually for flying or for swimming or for playing the piano. So once body parts exist and have established their identity, they are not necessarily tied to a particular function. And even that is sort of different, because certain parts don't change their function, like the heart has been a blood pumping organ, and I'm not aware of any example where the heart stopped being used for that purpose, right? So it's a question of how central and how well a particular part and function is integrated into the whole organismal context. And so that's the reason why I like to talk about character identity as a developmental morphological fact, and functional specialization as something that is attached to it, because a morphological and developmental identity can be more stable than functional role, right? So functional role is to some degree context dependent, basically how you use it. I mean, maybe a more dramatic example of change of function is the lung, right? So which we recognize as a respiratory organ in bony vertebrates. But once you have a lung and you fill it with air, and if you are an aquatic organism like the fishes, the very fact that you have air in these parts of the body also influences your buoyancy, right? Mm-hmm. So then if you are not depending on air breathing anymore, you can actually turn the lung into a buoyancy organ, which in fishes is called the swim bladder, right? And it completely loses its function for respiration, and now is only there for the purpose of regulating buoyancy. So that is another example. So swim bladder and lung genetically and developmentally is the same thing. It looks different, has different functions. Is another example of how the function of a body part can change in evolution.
Right. So in your work you talk about the concept of evolvability. Could you explain it?
Yeah, okay. You have hard questions for me.
Yeah, that's the point here.
So evolvability is is the idea, or originated from the idea that okay, let me start from the basics, right? So
Yeah.
the current model of evolutionary change is largely based on the idea that genetic changes can arise randomly in the genome, and they either have a negative effect, and selection will eliminate them on average, or if they have a beneficial effect, then natural selection may preserve them. It's not guaranteed, but there's a certain chance that sort of the better genes get conserved, and that's how adaptive evolution is happening. So how bodies become better in what they are doing. And now, then people had a brilliant idea and said, well, if this Darwinian process of mutation and selection is such a powerful process that it can produce, complex organisms like ourselves, why don't we use it in engineering, right? So why don't we make better machines by just mutating them and selecting the best machines, right? And that's actually something that people have started doing in the 1970s and 80s. And the intellectually interesting result of those was that it can work, but it doesn't always work. Maybe the most clear example are attempts to evolve computer programs by random mutation, random change, and selection. Just you test each program, you mute them, you run them on the computer. If they work or work better, then you maintain them, and then you mute it again. Now, what people learned when they did this is that whether that works or not critically depends on how random change is introduced into, let's say, the computer program or into some physical machine. Also, people also did this with physical machines like jets or airplane wing profiles and things like that. And it turned out that it is very critically depends on how this how the introduction of random change is done in order to have any chance to produce better outcomes, right? Which, if you come back to your question, it means that evolvability is not guaranteed by random change and selection. Random change needs to be done in the right way in order to have any chance of being successful. And evolvability is a term that sort of tries to measure or reflect the fact that some systems are better in producing better outcomes by random change than others. So that's, I think, the best way to introduce the intellectual history of that very idea. Now, whether and to what degree real organisms differ in their evolvability is still subject to vigorous research. And I think the problem for biologists to recognize that evolvability is an issue at all is that we usually, as zoologists or neontologists, so working with living animals, we are studying the winners of the evolutionary game. We usually don't know which organisms did not make it because that's the paleontology department that is studying those animals. And, admittedly, we don't know too much about these organisms. We don't know anything about their genetics. At the best, we can know something about the skeletons and their bodies, which is a lot, and this is important to know. But since we biologists or neontologists, as we should call ourselves, studying the winners of the evolutionary process, we don't even recognize that evolution may not happen efficiently because we only study the ones that did evolve efficiently, right? So it's only through the detour over evolutionary engineering and evolutionary computer science, it became clear that evolvability itself is an issue to understand. And in a way, it is the counterpart or the mirror image of constraints. Right? Constraints are the things that cannot easily evolve or decrease evolvability of certain parts of the body. And evolvability is the opposite. It's the opportunity to change or the ability to produce potentially adaptive and functional variants or mutations.
Mm-hmm. So you also study pregnancy. How did pregnancy evolve?
Well, so if we say pregnancy, of course we talk about many biologically quite different phenomena, right? Usually the term that is used is viviparity, meaning that a female gives rise to live young rather than laying eggs and then incubating them, and then the neonates or the hatchlings are getting out of the eggshell. And that is actually a phenomenon that happens quite often. In many animals have independently evolved the ability to give rise to live offspring. And if you look in particular, so most of the work on pregnancy, of course, is with mammals, because we are mammals, so we study ourselves and we study the reproduction of farm animals because that's commercially important.
Yeah.
And but the origin of pregnancy in mammals is a unique event, and so it's hard to know why it evolved in mammals. So our information about when it's preferred to evolve pregnancy comes from reptiles, so snakes and lizards and so on. And there it seems to be the case that populations that live in higher elevations and in colder climates are more likely to develop, to retain the eggs in their body rather than laying them and then incubating them outside. And so I think one of the driving factors in the evolution of intrauterine development is the ability to control the environment of the developing embryo more precisely than if you lay the egg and then either expose it to the environment or you have to regulate their environment by brooding on it and so on. So from an ecological evolutionary point of view, that's probably one of the driving factors that leads to evolution of pregnancy.
Mm-hmm. In the context of the origin or the evolutionary origin of pregnancy, you've studied endometrial stromal cells. So what are these and what was the role that they played here?
Right. So the endometrial stromal cell, so the decidual cell, is a cell that evolved in the context of the evolution of mammalian, or let's say placental pregnancy. And there we have to realize that mammalian pregnancy is quite special compared to most of the reptilian pregnancy. And so the reptilian, or in the simplest case, viviparity in reptiles simply consists of keeping the egg inside the mother and then let the hatchling either hatch inside the uterus and then it crawls out of the cloaca, or you lay the egg shortly before the animal is actually crawling out. This is, for instance, the case in a European lizard called Zootoca vivipara, where it's primarily egg retention that's happening. Now, a big difference in mammalian pregnancy, or let's say placental pregnancy, is that the separation between the embryo and the mother gets lost, so the eggshell gets removed, and the embryo in many cases deeply embeds into the tissue of the mother. So this is what's called embryo implantation, and this allows for a much more intimate relationship between the placenta, meaning the fetus, and the mother, and so the placental mammal also pregnancy is characterized by the development of those of placenta. Now, think about it from the point of view of the mother's organism or the mother's cells, right? So there is an embryo coming down the tubes and starts digging into the tissue of the mother. Now, from the standpoint of the cells of the mother, having this embryo there is what he or she is doing. The embryo is causing a wound, right? Mm-hmm. And having a wound, whether there's infection or not, naturally would lead to inflammation. And we know that the early stages of embryo implantation, even in humans, is actually a process that includes a lot of inflammatory processes. Now, so what we discovered a few years ago is that in the marsupials, so the sister group of mammals to the placental mammals, they have a very short pregnancy, and even within a 2-week pregnancy, they have a very short physical attachment between the fetus and the mother. It's about 2 days or 2 to 3 days. And during this attachment, the uterus is increasing progressively the expression of so-called inflammatory mediators, meaning genes that have to do with inflammation. While in the eutherians or the placental mammals, after this initial inflammation is actually turned off, and allows an extended gestation time. So basically, the mother allows the embryo to sustain a wound and maintain the wound open for the time of the pregnancy. Now, in order for that to happen, you have to change the way the tissue reacts to injury, at least as long as the embryo is there and the hormones are the right ones. And our results suggest that the residual cell evolved in order to allow this switch from the inflammatory reaction to the presence of an embryo to a state in which this open wound can be maintained. And in order for that to be possible, you need a cell or some kind of cellular interaction network that suppresses the inflammatory reaction. Because if inflammation could go on unbridled, it would lead to the destruction of the embryo, because there would be certain immune cells would come in and destroy the embryo, even in the absence of adaptive immune reaction. So our interpretation, our meaning, my collaborators and I think that the residual stromal cell evolved originally to downregulate the natural inflammatory reaction to the presence of an embryo, particularly an embryo that has the capability of embed itself into the tissue of the mother.
Mm-hmm. So that already touches a bit on my next question. Why is it that the fetus is not attacked by the mother's immune system?
Well, so I actually believe there are two different problems that we need to recognize. The one that I just explained, namely the question of why the so-called innate immune system that causes inflammation, regardless of whether a foreign body is recognized or not, is the regulation of inflammation. So to make clear that inflammation not necessarily has something to do with the rejection of a transplant or the rejection of an infection. Only think about an injury to your leg, that you break your leg or you sprain your ankle, that leads to an internal injury, not to infection, as long as the skin is not broken. But still, this internal injury leads to inflammatory reaction, right? So that's so it doesn't need to be an immunological rejection of a foreign body in order to get inflammation. So that kind of inflammation that we are talked about before is this sort of sterile inflammation that is triggered by a damage to the tissue. Okay, so this is the immediate thing that happens during the implantation phase, and if that wouldn't be regulated well, the innate immune system would attack the embryo with certain cell types that have the capability of just attacking whatever causes an injury. And then once this fetal-maternal relationship is established for long enough, then potentially the adaptive immune system of the mother could recognize that it's not only a tissue injury, but there's actually a tissue injury by a tissue that is not genetically the same as its own body. And that is the classical immunological paradox of pregnancy, pointed out or articulated by Medawar in 1953. And there has been a lot of research trying to figure out what's going on in the relationship between the mother and the fetus to prevent that. So one factor is, for instance, so all cells present in the body small fragments of their proteins on the surface, so that the immune cells can sort of taste them and see whether they belong to the body or not. And it is only if there's a mismatch between what the immune system expects and what they find on the surface of a cell, then the immune system starts to kick into action. And it turns out that the cells on the surface of the placenta, the ones that actually face the maternal bloodstream, are just not expressing these proteins that present the peptides to the so-called antigen recognizing cells. So this is one way. There are other mechanisms on top of it, because it has to be tightly regulated, and there's a lot of redundancy in there. So there are certain molecules produced that are suppressing the immune response. There's also evidence that the process by which so-called cytotoxic T cells, or the T cells that would kill cells that don't show the right identity, in order for them to get active, they have sort of a handshake they need to perform, and that is a so-called immune checkpoint. And there are various ways how this immune checkpoint can be inhibited, and that's also happening in the placenta. So it's a quite complicated and complex interaction network that evolved that prevents the maternal immune system to attack the placenta and thereby the fetus.
Mm-hmm. So just one last question. Is placentation in any way related to the development of malignant cancer?
that the short answer is yes, but the real answer is complicated, and that is for the following reason. So first of all, cancer and malignant cancer existed long before there was a placenta. Right? Every multicellular organism is vulnerable to developing cancer, because cancer essentially is the breakdown of the growth control of cells in the body. Right? To have a functioning multicellular body, every cell is supposed to do its job and nothing else. But there can be mutations to these cells, and then they escape this control, and that is what we usually call then a tumor. And if they grow out of control, then it kills the individual. And that's true for all multicellular organisms. Now, what can have changed once placentation in mammals has evolved is two things. The one is, and we discussed already, the ability of the placenta to escape the immune surveillance by the mother. And one theory says that, or a class of models says that once in placental mammals, the placenta has evolved mechanisms to prevent immune recognition or immune surveillance. These same genetic mechanisms can accidentally be turned on in a tumor. Mm-hmm. Then the tumor is no longer recognizable by the immune system, and then it escapes control, and then it's more likely to be fatal or malignant. There is a lot of sort of mechanistic evidence that supports that because what's going on in the fetal-maternal interface at the placenta also seems to be going on in the relationship between the host immune system and the tumor. The one question that has not been addressed in a systematic way is the question whether animals that have no placenta have less malignant cancers, because that if malignancy is helped by the genetic innovations that the placenta brought, that animals that don't have a placenta should be less vulnerable to malignancy, right? This has not been really carefully been investigated, but there's sort of anecdotal evidence that actually can be the case. For instance, it has been reported in the literature that it's very hard to cause malignant cancers in frogs. And of course, frogs are animals that never had a placenta. But that's only anecdotal. But it is at least a research question that in principle can be answered. Another aspect that is has to do with the evolution of placenta that we have researched in my group is the question of are different forms of placentas leading to different vulnerabilities in terms of malignancy. And for that, one has to recognize that in the placental mammals, the ancestral placental mammal had a deeply invasive placenta, so one that deeply embeds into the tissue of the mother. But there are quite a number of so-called placental mammals where the placenta does not invade, like the cow and the tupaya and the pig and the horse and so on. So those are animals where the placenta develops, but it doesn't invade into the tissue of the mother. Given the evolutionary evidence, our conclusion is not but the community's conclusion is that these non-invasive placentations are actually innovations, are new inventions, where, as it turns out, most likely the mother evolved and the capability to keep the fetus out of its tissue. right? And we know that because if you take an embryo outside its context and let it develop in the, let's say, the body cavity, in the pig, for instance, it's still invasive, but it's not invasive in the uterus, which means the uterus is the tissue that keeps the fetus out, or prevents the fetus from invading, from embedding. And what we found is that in fact animals where the mother evolved a way to keep the placenta out are also less vulnerable to cancer spread. So there's a quite impressive case report out of India, and it has to be out of India because it has to do with cows, and in the US and probably also in Europe, cows don't get old; they just get slaughtered in a young age, and only in India they are actually getting rather old. And there's a case report out of India that shows a cow with a very large melanoma-like lesion, about a tumor of about maybe a kilogram or something like this, but no metastatic cancer in other parts of the body. While in a human, if a melanemic injury is about 4 millimeters deep, the survival rate is about 50%. If not treated aggressively, right? So there's very different, very large differences in the likelihood that a tumor turns malignant, and that depends on how well the surrounding tissue can sort of constrain the tumor from getting out of hand. Mm-hmm. And it looks like the evolution of a uterus that can keep the fetus out of its tissue has so-called pleiotropic effects. I mean, coming back to this concept again, on the tissues of the rest of the body, and these tissues, let's say in a cow, are more able to restrict the migration of tumor cells. So we have shown this experimentally, and we even could show that we can make human cells more resistant to invasion from cancer cells if we change the gene expression in these human cells to be more like cow cells. So which means that we can actually identify which genes are responsible for this ability to contain the spread of a cancer. And our hope is, by understanding how the cow is doing it, we can eventually help the human body to prevent the spread of cancer in ways that I cannot imagine right now, but that's the general hope of this line of research.
Great. Okay, so Dr. Wagner, just before we go, where can people find your work on the internet?
Well, I think it's the easiest is to just search for my name in the biomedical database, NCBI or PubMed website, or on my lab website at Yale University, and much of it should be freely available online.
Okay, very well. So thank you so much for taking the time to come on the show. It's been a real pleasure to talk to you.
Thank you so much for having me. It was a great pleasure talking to you about these things. Thank you.
Hi guys, thank you for watching this interview until the end. I would like to ask you to please consider supporting the channel. You will find links to Patreon and PayPal in the description box of this interview. Otherwise, and if you like what I'm doing, please share the interview, leave a like and hit the subscription button. This show is brought to you by Enlights, Learning and Development Done Differently. Check their website at enlights.com. I would also like to give a huge thank you to my main patrons and PayPal supporters, Karen Litzke and Blanchett, Pereira Larsen, Lau Guerrero, Francis Ford, Ernst Frederick Sunda, Ricardo Vladimiro, Craig Healy, Adam Kessel, Olaf Alex, Jonathan Wiesel, Jacob Klinkby, Matthew Whittingbird, Arno Wolf, Tim Hollesey, Henrik Alenia, John Connors, Paulina Barron, Philip Force Connolly, Jerry Mueller, Herbert Gintis, Rutger Voss, Bo Weingard, Rebecca Newberger Goldstein, Dan Demetrio, Robert Windager, Rui Inácio, Arthur Coe, Zup, Marco Neves, Colin Holbrook, Susan Pinker, Bernardo Seixas, Pablo Santurbano, Simon Colomba, Jorge Espinha, Phil Kavanagh, Corey Clark, Mark Blythe, Roberto Inguanzo, Michael Stormir, Eric Neumann, Samuel Andreev, Tiago Nunes, Bernard Hughney, Alexander Dunbauer, Omri Hickson, Fergal Cusson, Evan Bodrenko, Al Herzog, Don Ross, Jonathan Librant, Oslin Bullitt, Nathan Nguyen, Stanton T, Samuel Correa, Eric Hines, Mark Smith, J.W., João Eira, Tom Hummel, David Sloan Wilson, Yacila Dez Araújo, Eden Solon, Romain Roach, Dmitry Grigoriev, Diego Antonio Correa, Tom Roth, Yannick Punter, Adana Rusmani, Charlotte Bliss, Nicole Barbaro, Adam Hunt, Pavel Ostazewski, Nelek Bach, Catherine and Patrick Tobin, Al Ortiz, Guy Madison, Gary G. Hellman, João Linhares, Lida Cosmides, Saima Afzal, Adrian Yegi, Nick Golden, Paulo Tolentino, João Barbosa, Jules Price, Edward Hall, Edin Bronner, Franca Bortolotti, Gabriel Pons Cortez. My producers is our web, James Frank, Lucas Stafiniak, Ian Gilligan, Luís Caetano, Tom Vanek, Dam Curtis Dixon, João Linhares, Benedict Mueller, Vega Guidi, Sardus France, and Thomas Trumbull, and my executive producers Michel Rugieski, Rosie, James Pratt, Matthew Lavender, Sergio Quadriano, and Jason Party. Thank you for all.